salinity stress. So when the soil is saline, it causes the salinity stress, and this salinity stress, it affects the nutritional status of the plant. Some ions are very high in the plant, some ions are less in the plant. Okay, so in salinity stress, solute or ion concentration are very high. Okay, so when solute and ion concentration are very high, it reduces the osmotic potential. Okay, so more the solute concentration, the more negative value will be of the osmotic potential. So when the osmotic potential is reduced, it also reduces water potential. Okay, so when a water potential is reduced, that means this cell lacks water. Okay, so see, when there is imbalance in the mineral content of the soil, it causes salinity stress, which affects the nutritional status of the plant. So due to this high solute concentration near the rooting zone, it generates a low osmotic potential. So when the osmotic potential, which is designated as psi s, when this is low, the water potential, which is designated as psi w. So when osmotic potential is low, the water potential is also low. When water potential becomes low, which means that this cell has very less water. Now this salt injury involves both osmotic effects and specific ion effects. Okay, so specific ion toxicity. So I would like to say here that salinity stress, can be specific or non-specific. It can be specific or it can be non-specific. So when it is non-specific, this salinity stress causes osmotic stress. So when there is osmotic stress, the water content is reduced. Okay, when there is osmotic stress, it causes water deficit. This we already discussed when we were doing water stress. Okay, high amount of solute reduces the availability of water. Now specific stress is what it when the concentration of ions or specific ions are accumulated. So when the concentration of specific ions are very high, it leads to the toxicity. So specific stress is due to the accumulation of specific ions that lead to toxicity. Okay. So see here, specific ion toxicity effects occur when injurious concentration of ions, particularly sodium, chloride, and sulfate, accumulate in the cells. So these are more injurious than the other. So whenever sodium concentration is very high, it not only injure plants, it has several other effects that it also degrades the soil structure. It decreases the porosity and water permeability. Okay, so an abnormal ratio of sodium to potassium and high concentration of total salts, they altogether inactivate several enzymes and they inhibit protein synthesis. Okay, so in this way, the general water balance of the plant is affected. Okay, so as these all ions are when accumulated, they decrease the water potential. When they decrease the water potential, the water balance of the plant is affected, because to maintain the balance now, to gain the water again, what leaves need to do? They need even lower water potential to maintain the downhill gradient of water. Now salinity depresses growth and photosynthesis in sensitive species. So some plants will tolerate the saline condition and other will be sensitive to it. So there are two kinds of plants, halophytes and glycophytes. So halophytes are the plants which are native to the soil, saline soils. They grow in the saline soils and they complete their life cycle in that environment. But glycophytes, called as sweet plants, they have high amount of sugar in them. They are non-halophytes and they are not able to resist salts to the same degree as halophytes. They resist but to a very less degree. Okay, there is a threshold concentration of salt which they can tolerate, beyond which they begin to show the signs of growth inhibition, leaf discoloration and loss of dry weight. So it can be said that halophytes have a greater capacity than glycophytes for ion accumulation in shoot cells. Okay, this how they accumulate the ions in the shoot cells we'll see. So see, this graph is showing that how the halophytes and non-halophytes are grown in response to the high concentration of chloride ion. See, just to compare. Okay, so with group 1A, this one. So group 1A includes sea blight and salt bush. These species show growth stimulation with chloride leaves. So these 1A are halophytes which are highly tolerant to the salt. Okay. Group 1B, they include Townsend cordgrass and sugar beet. These plants are tolerate. They can tolerate. They are also a type of halophytes. So they are highly tolerant to the salinity stress. These halophytes are also tolerant, but their growth is retarded. See, in the third case, it includes both halophytes and non-halophytes. It includes salt-tolerant halophytic grass which lack salt gland. So these are also halophytes. I'm labeling it here. Okay, I'll just write lack salt glands. So group 2 lack salt glands. Okay. Number 3, this species are very much sensitive to the salt concentration. So number 3 are the non-halophytes. Okay, they are very sensitive to the salt concentration. See, their growth rate is so less in response to the very less concentration of chloride ion. Clear? So non-halophytes include citrus, avocado, stone fruit, etc. So this was a question once came in CSIR NET. So shown below is a graph representing the growth of different plant species subjected to salinity relative to that unsalinized control. Which of the following statement is not true? So plants in group 1A are extreme halophytes while very salt sensitive species will be the part of group 3. Okay, so group 3 is very salt sensitive. Group 3 are non-halophytes, very salt sensitive. So one is saying that, and one A is saying that they are extreme halophytes, that they can tolerate high amount of salt concentration. So number A option is perfectly correct. Plants in group 1A are very salt sensitive. No, if A option is correct, then this is wrong. Halophytes which can tolerate salt but their growth is retarded will be a part of group 1B. Yes. So these are also halophytes but their growth is retarded. Yes, just now we studied. So this is also correct. Non-halophytes which are salt tolerant but lacks salt glands will be a part of group 2. So these number 2 are, this includes both halophytes and non-halophytes. Okay, they lack salt gland. Which lack salt gland. And this group 2 includes both halophyte and non-halophyte. So this is also correct. So we found that number B is the incorrect one. This is our right answer. Next, different strategies to avoid salt injury. So what plants do? So when the salt, when the ions are solute, when they accumulate, so different strategies to avoid salt injury. Now plant minimize this salt injury by excluding the salt from the shoots and from the leaves, from the meristems of the shoots and the leaves, and these regions are the actively growing and expanding regions, and these are the actively photosynthesizing tissues of the plant. Now there are some plants which are salt sensitive. They also are able to show the resistance to the moderate level of salinity. How do they show this resistance? That they prevent the transport of harmful ions to the shoot. Now how this prevention occurs? So see here that this is epidermis. From the epidermis the root hair cell come out. This is epidermis, this is plasmodesmatal connection. Okay. After epidermis it is the cortex, which will also have plasmodesmatal connection. After cortex it is endodermis. Then after endodermis it will be pericycle. Then after pericycle it is the xylem vessel. Okay. So see, this is the region of the cell wall. The movement through cell wall is known as apoplastic transport, and the movement inside the cell is known as symplastic transport. Okay, so the third one is endodermis. Now, in the apoplastic region of the endodermis, there is the presence of a strip known as Casparian strip. Look here. In the apoplastic region of the endodermis, there is the presence of strip known as Casparian strip, and this strip is water impermeable. Okay. Now, water transports both apoplastically as well as symplastically. Water will be transported through the cell wall also, and it will be transported from cell to cell also. Okay, but when they reach the endodermis, since the Casparian strip is present, which is water impermeable, they cannot pass through the endodermis. They have to enter the symplastic pathway. This pathway has to enter the symplastic pathway. Okay, once they are near the endodermis, so after they cross the endodermis, they can again move to the apoplastic pathway or the symplastic pathway. Okay, there are plasmodesmatal connections in between everything. So what happens once they enter inside the cell? Once the ions are inside the cell, the plant or the cell shows the mechanism where they concentrate the ions inside the vacuoles. Okay, they're able to concentrate the toxic ions inside the vacuoles, which will help the plants to minimize the concentration of different ions in the cell. Okay, so when the ions increases in the cell, that will be very harmful because they shows very adverse effect on the plant. So once the ions are inside the cell, they are the like their availability is minimized by concentrating them into vacuoles. Okay, so in this way, see, to bypass the Casparian strip, ion needs to move from the apoplast to the symplastic pathway across the cell membrane. Now this transition offers salt resistant plant a mechanism to partially exclude harmful ions. Okay. Next we discussed that in group 2, halophytes and non-halophytes, they lack salt glands. So we need to know, there must be curiosity in the mind, that what are salt glands? So some salt resistant plants such as salt cedar and salt bush, they have salt glands on the surface of their leaves. So these ions, when once they are inside the cell, they are transported to these glands. Now they are once when transported to these glands, the salt in these glands crystallizes and they are no longer harmful. Oxygen may protect during acclimation to various stress. So how oxygen protects? So these oxygen acts as a electron acceptor. These oxygen acts as an alternative electron acceptor. Okay, they are also able to accept the electron like NADP. Okay, so whenever there is like the production of high energy in the plant, when the plant cells are highly energized in the condition, say for example high temperature, high light intensity, then what happens? That the plant cells become charged, the plant cells becomes excited. So when the cells are excited, they need to transfer the electron to another cell to dissipate the energy, otherwise their system will be damaged. To dissipate the energy, one way is to transfer the electrons to oxygen. So once oxygen accepts the electron, it is converted to reactive oxygen species. Okay, so look here. So here oxygen is acting as an alternative electron acceptor for photosynthetic electron transport also. Okay, in photosynthesis electron transport, if you remember that there was a Z scheme. PS1, PS2 becomes excited. Just writing few proteins. It becomes excited through the several of the proteins in uh, the intermediate. It is transferred to PS1. Then PS1 helps in the formation of NADPH. Okay, so instead of NADP acting as an electron acceptor, it can be oxygen also. Now, once oxygen is reduced by PS1, it is known as MEHLER reaction. So, instead of NADP, when it is oxygen, so when electrons are transferred from PS1 to oxygen, this is known as MEHLER'S reaction. Okay, so once the electrons are received by oxygen, it forms what we all know, that it forms the superoxide radical. It forms a reactive oxygen species known as superoxide radical. Okay, it will form O2 minus. Clear? So it will form superoxide radical, which is symbolized as this. Okay, next. Now, once this superoxide radical is formed, what happens? That these superoxide radical must be removed by the enzyme superoxide dismutase. Okay, so when the superoxide are formed, they are rapidly scavenged. Scavenge means converted into non-toxic products. They are the superoxide is a toxic product. They are quickly converted into non-toxic product. The superoxide is converted into, this is superoxide, it is converted into non-toxic product, which is hydrogen peroxide. So here this is a scavenging reaction, and the enzyme required here is superoxide dismutase, which is present in several cellular compartments including chloroplast. So this superoxide dismutase is able to scavenge the superoxide radical, okay, by forming the non-toxic product, which is hydrogen peroxide. So this is somewhat non-toxic. This is not totally non-toxic. This is somewhat non-toxic. Now, what is radical? Radical means it has an unpaired electron. The ones which has unpaired electron are very reactive. Okay, it has an unpaired electron. Okay. Now, once H2O2 is formed, once hydrogen peroxide is formed, it needs to be reduced to water. Okay, in the chloroplast itself by sequential reduction with ascorbate, glutathione and NADPH. Very, very important to remember. Okay, so what happens? This H2O2 will be reduced to water by the action of ascorbate glutathione and NADPH. I'll show you how this happens. Now what is the necessity of converting the H2O2 into water? So if this hydrogen peroxide is not converted to water, then this H2O2 may form hydroxyl radical. Okay, this H2O2 will react with superoxide to form hydroxyl radical. It will react with superoxide radical to form hydroxyl radical. Now hydroxyl radical is the most dangerous radical. So once it is formed, it damages many of the proteins very quickly. Okay, now this pathway for the protection of ROS, that is conversion of conversion of SOD to H2O2 and then H2O2 to water. Okay, this conversion is known as Asada Halliwell Pathway. So see here in this picture, the TS1 is giving the electrons